Artelo Biosciences is developing a next-generation cancer therapeutic based on the science of lipid signaling pathway modulation. Lipid signaling is a continually occurring essential process in human cells and involves fatty acids in a complex molecular chain of events. Emerging data indicate that in many cancers, including breast and prostate, excess lipids and their signaling within cells contribute to disease progression. Artelo's lipid signaling modulation platform has the potential to be the next revolution in cancer therapeutics. Fatty acids are hydrophobic, or water-fearing, and need to be chaperoned within the intracellular environment. Specialized proteins called fatty acid binding proteins, or FABPs, act as the chaperones for these fatty acids, allowing their transport and interactions within the cell. Fatty acid binding protein 5 is found in epithelial or barrier cells throughout the body. One of the roles of fatty acid binding protein 5 is to regulate cell growth, and it has been found to be overexpressed in many cancers, including prostate cancer, feeding the cancer cells with excess fatty acids and encouraging tumor growth and migration. A cancer patient with high levels of fatty acid binding protein 5 in their tumor is less likely to survive the cancer. How exactly do fatty acid binding proteins encourage tumor growth and migration? Excess fatty acids bind to fatty acid binding protein 5 inside the cell and are transported to nuclear receptors. Once within the nucleus, they increase the production of proteins that support tumor growth and migration. These proteins increase cancer cell division and survival, stimulate the development of new blood supply to the tumor, and encourage the migration of cancer cells to other parts of the body. If this pathway can be inhibited, then these cancers may have their growth slowed or even stopped. Artelo's research team is currently working with scientists at Stony Brook University, who are pioneers in fatty acid binding protein 5 inhibitors to advance the lipid signaling program towards first-in-human trials. At Artelo, this program is called ART26.12. In laboratory studies, ART26.12 was shown to decrease tumor size in mice injected with prostate cancer cells. Importantly, ART26.12 also decreased the spread of the cancer to other areas, such as the liver and lungs. Another potential benefit of ART26.12 may be its ability to work synergistically with standard of care chemotherapy drugs called taxanes. Taxanes are traditionally used to treat metastatic prostate cancer. However, prostate cancer can become resistant to chemotherapy, and there are many side effects of taxanes. Data from Stony Brook University show that fatty acid binding protein 5 inhibitors do not interfere with the anti-tumor actions of taxanes, but instead are able to work synergistically to cause even greater reductions in tumor growth than with either drug alone. Combining ART26.12 with lower doses of taxanes may result in fewer side effects and a more tolerable experience for people treated for prostate cancer. Given the encouraging anti-tumor and anti-metastatic non-clinical results observed with ART26.12 and the demonstrated synergistic potential with standard therapies, ART26.12 is expected to have broad application in certain vascular-driven malignancies such as prostate, breast, and cervical cancer. Artelo is advancing research with ART26.12 for multiple potential applications. With ART26.12, Artelo Biosciences is leading the way in the new frontier of next-generation cancer therapeutics. For more detailed information about Artelo Biosciences and its development programs, please visit artelobio.com.